to hear about what's going on in Baffin Bay. So what I'm going to share with you tonight, and please ask questions throughout. If you have a question, just shout it out or raise your hand. What I'm going to share with you tonight is a story that we've been working on for Baffin Bay for almost 4 years now. This is a project that was funded by the EPA, and we've got a lot of partners involved in the project. It's primarily it's a project at A&M Corpus Christi, but we have a lot of partners, including some that are referenced here. We're working with the Heart Research Institute, Texas Water Resources Institute, and also UT Health. So we've got a lot of partners in this, and maybe to start, let's see if this will work. Well, it was working so well earlier. Of course it doesn't work now. Okay. We are frozen for some reason. Okay, there we go. That works. We can do it that way. It woke up. Oh. All right. So to start, I wanted to share this beautiful photo of Baffin Bay. So I don't know who took this photo. Dominic, was it you?
It was me.
All right. I was hoping you'd be here so I could give you credit for it. I figured it was you or Edgar, right? It's a really good photo, and I think it really captures the beauty of the bay. So Baffin Bay is frequently referred to as the jewel of the Texas coast, and that's a really appropriate term, right? It's a beautiful bay, and it's known for sport fishing, right? And anglers love to go to this place. It's a fabulous place. It's also known for these really cool circlet reefs that are part of the ecosystem there. That's part of the reason why the fish are there. It's part of the reason why the anglers like to go there, right? So it's this beautiful place. And if you look on the news about Baffin Bay, you'll see that there's a lot going on down in Baffin Bay. There's this really cool project in Baffin. It's called Baffin Back, and it's work that Mike and others started more than a decade ago that has been captured and magnified by this Texans by Nature conservation wrangler program, with this attempt to bring Baffin back to its natural status, right? And there's work going on to save these circulid worms that are part of these really cool reefs that are an important part of the ecosystem there. I love Texas Monthly. That's a really good article. Please check it out. And the Caller Times just did a 2-part series about a lot of the work that's going into Baffin Bay too. It came out this past week, right? So check that out as well. And again, about 4 years ago, we got this award from the EPA to take a look at what's going on in Baffin Bay with bacterial pollution, right? And the rationale for the project was that 3 of the tributaries in Baffin, Petronilla Creek, San Fernando Creek, Los Olmos Creek, they're all listed, different water segments in those tributaries are listed on the Texas 303D list for impaired waters because of elevated bacterial levels. And Los Olmos Creek was one of the more recent listings, but it was listed just this past year. The others have been on there for a little longer. And routine water sampling has produced bacterial concentrations that are many times above where they should be, right? So in the numbers of thousands rather than double digits, right? So bacterial pollution is a problem there. That's what I'm talking about tonight, bacterial pollution. There's a lot of work going on in Baffin Bay regarding other pollution, like nutrient pollution or chemical pollution, but bacterial pollution is what I'm focusing on this evening. And the bay is also showing signs of eutrophication. So there's elevated dissolved organic carbon, and there's elevated organic nitrogen, elevated chlorophyll, there's hypoxia events, dead zones, and there's harmful algal blooms, the brown tides, the Oriumbra agulensis, right? So, and a lot of that work has been conducted out of the Heart Research Institute and Dr. Mike Wetz's lab, right? So you can just Google Scholar him, and you can find a lot of the work that's been done in Baffin Bay. But the bacterial work is kind of new. So this is a screenshot of what was on the EPA website in their 22 annual report. This project was featured in that report because of how collaborative it was. So A&M Corpus is the lead, but we have a lot of partners in this. There's 8 of them listed here, again, including the HART, the Nueces River Authority, the Water Utility Lab for Nueces County, the Physical and Environmental Sciences Lab with Corpus Christi, the Center for Coastal Studies with Corpus Christi, UT Health, the Texas Water Resources Institute, and Voices of the Colonials. This is a really cool project. There's a lot of partners involved in this. Building and managing the subawards has been a nightmare, but it's really cool when all this stuff comes together. And it's been a lot of fun to work with these people. But the purpose of the project is, as highlighted here, to identify fecal pollution sources and their associated health risks in Baffin Bay. So there's 2 parts: identify the fecal pollution sources and then quantify the health risks. So that first part, identify the fecal pollution sources, that's microbial source tracking. That's what my lab does. The second part, the quantification of risk, I don't do that. That's something that Anna Gitter with UT Health does. So that's part 2. I won't be talking about part 2 tonight. Hopefully we can have her here in the future maybe to talk about part 2. But here's a list specifically of some of the contributors. So people that are actively working in watersheds to contribute to this project. People like Dr. Wetz, Dominic, who's here, Sam, who's here, and Athena, who's here. Then we've got the microbial source tracking department. That's my lab. So Dr. Nicole Powers, she's not here because she lives in Dallas, but she is the expert on coastal bacterial pollution in the state of Texas. She's the person, right? And we were really fortunate to have her on this project. It's really elevated work. Nora Bleth, who's a PhD student in the lab right now. So a lot of this data that I'm going to show you today, it's data that Nora produced in the lab and Nicole helped analyze, right? And we also have undergrads like Jacqueline, who's participated in the project. And eventually we'll have this quantitative microbial risk assessment. It's one thing to know the numbers of bacteria, but we need to know what that means. What kind of risk does that pose us if we're swimming or if we're fishing, right? So that's the next part. And then we have outreach components. There was work that we did with Ana Garcia with Voices of the Colonials and Aaron Hill with the Center for Coastal Studies. And Dr. Lucas Gregory, he developed the watershed protection plan for Baffin Bay, and what and the watershed protection plan predates this study. What he's doing is he's evaluating and prioritizing different best management practices as we get this data, right? He's our guy on that. And here are some beautiful, happy faces. So we have Ana Garcia here with Aaron Hill at an outreach event. Nicole Powers, Dr. Nicole Powers is right here, and Nora Bleth is right here. Again, the data that I'm sharing tonight is primarily theirs. I am going to share some data from Dr. Lucas Gregory too about land use and wildlife and livestock assessments. Lucas is right here. He's with the Texas Water Resources Institute. Dom, I got your beautiful face up here. We got Anna Gitter up here in the corner. Dr. Gitter is going to be doing our QMRA. Dr. Mike Wetz over here. Sam is our guy on the boat. He's our Uber. We've got undergrads here, Jacqueline Nicolay, Andrew Johnson, and there's others that have worked on this project too. This is the part that I really have fun with. Again, I get to work with all these really brilliant people. That's the thing I like most about my job. All right, so let's dive right in and talk about where this work is conducted. It's conducted in Baffin Bay. We did this, we did 18 monthly collection trips. We had 6 sites on the bay. It might be a little hard to see them, but we have 6 bay sites right here. We have 1 site on Los Olmos Creek, although it's pretty much a bay site, it's tidal. We have 3 sites on San Fernando, and we have 2 sites on the Petronilla. So equally divided 6 bay sites, 6 tributary sites. Dominic had a team that went out to sample the bay sites, and Sam had a team that went out to sample the tributary sites. And it worked like a well-oiled machine. It never failed. It was perfect for 18 months. And we got a lot of samples out of it, 216 samples. And we collected gobs of data. The data that I'm talking to you about tonight, though, I'm going to talk to you about fecal indicator bacterial data, E. coli and enterococci. And I'm talk to you tonight about microbial source tracking data, which we did microbial source tracking for humans, cows, pigs, and gulls. And those are just the abbreviations for the molecular markers for those detections. How many of you remember going to get a COVID test at CVS, where you had to do the PCR test, right? The rapid ELISA test wasn't good enough, right? The home kit test wasn't good enough. You had to go to CVS to get the PCR test, and you get your results the next day. Well, the test that they use, it's a PCR test, polymerase chain reaction. It's based upon a genetic marker that's in the COVID virus that they detect and quantify using PCR, right? And that's exactly what we do with these markers. These are markers that are associated with these different animal hosts, and we quantify them with PCR. It's the same thing as the COVID test. It's just a different marker. And it tells us what the sources and magnitude of the fecal pollution is. That's how we learn who's contributing more fecal pollution than somebody else. All right. So a couple quick terms here. Fecal indicator bacteria. This is what we've been using for decades. So fecal indicator bacteria include E. coli and enterococci. E. coli is the acceptable standard for freshwater, and enterococci is the acceptable standard for seawater. And it was the Coastal Health Beach Act of 2000 that required states and territories to adopt water quality standards that were protective of human health. Most states use these two. Some states, for instance, Hawaii, they have more. But enterococci is the standard that the Texas General Land Office uses for their beach watch program. And these are good things, right? Microbial source tracking is the next generation of detection. Because knowing there's fecal pollution, it's great if you know them. But if you don't know what the sources are, it's hard to mitigate it, right? You need to know the sources so you can focus on the problem. And that's what we're doing today. All right, so a little bit of history about fecal indicator bacteria. They indicate fecal pollution. They themselves may or may not be harmful to you. They may be harmless, but if they're present, they're an indicator that fecal pollution is there. And if that fecal pollution is human fecal pollution, it could compromise your health, right? There could be viruses, there could be bacteria, there could be protozoan parasites. Think Giardia or Cryptosporidium. That could make you really sick, right? So it's an indicator of fecal pollution. It really doesn't speak toward risk, though. It's approved by the EPA. It's not host specific. Remember, it cannot identify who the poop came from. And this concept of FIBs was developed back in the mid to late 1800s. This is an old concept. There was some methods development that happened in the 50s that led to this really cool membrane filtration technique. And that's the technique I was taught in grad school. But then in the 70s, they came out with this quantitrade method that was a little more high throughput, a little easier to do. And that's what we use today. So the test for E. coli and enterococci, it was developed, the method was developed in the 1970s. And just let that sink in. 50 years ago, right? That's an old test, right? How many of you are driving a car that's 50 years old? Right? How many of you want to base your health risk on a test that's 50 years old? No, you don't. Or at least you don't want it to be based solely on that data, right? You want more data. You want moderate data. And these fecal indicator bacteria, what you often see in textbooks is that there's a linear relationship between the number of illnesses and the concentration of the fecal indicator bacteria. It rarely works that way. I call it a purported correlation. Because if you go to a textbook, you'll see this, and if you go to the company that makes InterAlert and ColiAlert, the test, they'll tell you this too. But it rarely works that way. Usually, if there's a correlation, it's a very weak one. Sometimes it's no correlation at all. I'd say most of the time it's no correlation at all. And sometimes you actually get an inverse correlation, where the number of illnesses go up when the fecal indicator bacteria is the lowest, right? So again, 50-year-old test, it's got problems. Sometimes it works, sometimes it doesn't. But it's easy to do. It's an easy test. It's an affordable test. I call it the test of minimal effort. And we do that test in my lab. And I'll share with you tonight some of the data that we've got from the test. And we'll talk about, the pros and cons of it, and we'll talk about why we need microbial source tracking to kind of rethink and, I guess, shift the narrative about what we've been thinking about Baffin Bay. All right, here's that quantitrade test. There's one called ColiAlert, which does E. coli. There's one called InterAlert that does enterococci. They're developed by IDEX Laboratories. It's a $1 billion industry. You can buy shares. I wish I had known that they'd be the national standard back in the 70s. But it is what it is. It's a cool test. And again, it's affordable. It's easy. If you're going to have, dozens of labs all across the coast who are testing for fecal pollution, this is a good test to use because, you can contract with all those labs and they can all run this test with high confidence. But interpretation of the data, you have to be careful. All right. So, for instance, Texas Beach Watch, for their Texas Beach Watch program, they have 66 beaches that they monitor using this test. National Surfrider Foundation, they have 56 labs that are monitoring, 567 beaches using this test too. So there's good things about it, but we need to layer into it more modern testing, microbial source tracking. All right, and microbial source tracking, again, this is the COVID test, right? Same thing. And this is where we get to say, okay, who are the sources of pollution in a watershed, right? Wildlife can certainly be a contributor. And humans can certainly be a contributor. And livestock can certainly be a contributor. And I want to make clear that I'm not using this presentation to point any fingers. There's a lot of different sources of fecal pollution in the watershed. We have a much better idea who the contributors are, but any mitigation aimed at nutrient sources or any fecal pollution sources, it brings down everything, right? These mitigation techniques that we can use can ameliorate all this stuff collectively. So I'm not suggesting we just focus singularly on any one thing, right? But the ones we looked for in this study were human, cow, pig, and goat. But there's a lot of other contributors as well. And this is Nora, up in the layer. We have a lab on the 3rd floor that we call the layer. And this is where Nora does her best work. And she's quantifying these different host-associated markers. All right, what are some potential drivers of fecal pollution? Well, the quantity of fecal pollution can vary with temperature. When it gets warmer, those indicator bacteria, those fecal indicator bacteria, will grow better in the environment, right? Salinity, if you have a real high salinity, fecal indicator bacteria that came from inside you or inside a mammal, will die off more quickly in high salinity. Sea level rise, if you have inundation of septic fields, right, then that could potentially increase coastal bacterial pollution. Rainfall can increase fecal pollution from non-point sources. Human populations, right? So if you have a very large human population, there could be a correlation with population size. And if you have a large wildlife population or a large livestock population, that can also lead to an increase in bacterial pollution. And a lot of times when we talk about bacterial pollution, and I teach classes on this, and the students will all automatically go to wastewater treatment plants and septic systems. And I say, okay, that's fine, but if they're operating like they're supposed to, they're part of the solution, right? If a wastewater treatment plant or a septic system are doing their job, they're not part of the problem. That's not your source, right? And for the most part, these systems are doing their job. Sure, there's some aging infrastructure, there's some problems, especially with heavy rainfall or something. There's some septic systems that have failed, but by and large, these systems do the work they're supposed to do, right? I hate that I always get blamed, right? They do really good work. All right, and this figure here is something I pulled from one of Dr. Nicole Power's papers. She did an analysis of a 10-year data set from Texas Beach Watch to look at hotspots of fecal pollution across the Texas coast. And, she showed that this area here, Nueces and Kleberg, is a hotspot. And that includes Baffin, some parts of Baffin, right? Matagorn and Brazoria have a bigger problem, right? And we can talk about that at another time, but we've got some data there too. And Houston, of course, is a huge population center.
Other types of pollution that knock out these fecal volumes, we had metals as well as sewage, when you have low bacteria numbers.
Metals are toxic. Heavy metals are toxic.
Like oil spill plus sewage, all of a sudden your vehicles go down, but it's all to really just get to kill everything.
It could. It definitely could. And for people who are familiar with sediment studies in this area of Texas, South Texas, Coastal Bend, Baffin Bay, there are heavy metals naturally, right? So heavy metals are toxic to bacteria just like they're toxic to us. What's really interesting though, and I'm going on a tangent here, is that the same mechanisms that bacteria use to detoxify heavy metals, it's the same mechanism they use to detoxify antibiotics. So if you have heavy metal pollution, the bacteria from there will be drug resistant because they've been exposed to heavy metals. It's really cool. And Nora, the second chapter of her dissertation is on the resistome of Baffin Bay. So looking at all the antibiotic resistance genes, which is going to be super cool. And she's working with Del Mar undergrad right now, who's quantifying some of that. That's going to be a new story, and I'm super excited about that. Yeah, that's a good question. All right, we couldn't wait to get out in the field. We had to do a QAP, Quality Assurance Project Plan. Some of you know what I'm talking about. It was miserable. We did it, and then they finally let us out in the field, and we had a great time. Some of these photos Sam took. So we got a couple of the tributaries here. Here's the bridge that goes over Los Olmos Creek, and here's Nora and Bella out on their boat Uber, collecting samples. So we had a lot of fun. 18 sampling events between 22 and 24, and 216 water samples. Question? It wasn't part of the study. The NOFO, the Notice of Funding Opportunity from the EPA, it was a surface water study, right? So, but sampling the groundwater would be great. But, you know, for whatever reason, the way these studies work out, you'll have a project that's funded to sample surface water, and, you know, then there's an independent project that somebody gets funded to do groundwater. But they're never, like, done together, or they're rarely done together. But that's really valuable, right, when they get done together. It's also really valuable when you have independent projects, and then the data supports each other too. So, you know, there's different ways to do it. Either way is good, but yeah, groundwater would be nice. All right, and here's some of our results. This is the results for the fecal indicator bacteria. So this figure here shows the concentration of fecal indicator bacteria, and the units are most probable number per 100 milliliter. And basically think of that as a single bacterial cell. So if you got 1 MPN per 100 milliliters, that's a single bacteria, right? And we got Baffin Bay here, our 6 sites in Baffin Bay. We got our 1 site in Los Olmos Creek. We have our 2 sites on Petronilla and our 3 sites on San Fernando. And I don't think I mentioned this earlier, but E. coli is the acceptable standard for freshwater, and enterococci is the acceptable standard for marine water. So in the bay and in Los Olmos, because it's a tidal section of the creek, we use enterococcus, and in the tributaries further up, we use E. coli. And when I first looked at this, I thought, oh, this is really cool. You know, this mirrors the narrative that I've always heard about Baffin Bay, fecal pollution. You know, the thought is that Petronilla has a really big problem, right? That it's the problem child of the tributaries, followed by the San Fernando, and then Los Olmos is kind of just weird. It's this, it's hypersaline. It's a hypersaline tributary for quite a ways, and we've only got 1 site for Los Olmos because access is limited. But it's just an odd site, and it's an odd tributary. And then Baffin Bay proper, where we have 6 sites. You know, so you've got these tributaries coming in. These freshwater inflows are carrying high concentrations of bacterial pollution, but once they get in the bay, they dilute out. But the problem chiefly is, you know, the narrative is that Petronilla is the problem child, and things get diluted out in the bay. And the more time I spent with this, I was like, well, wait a minute, that doesn't make sense to me. And I started to look at the data a little more closely. And I want to describe to you some problems with this. And we'll start here with Baffin. All right, so we have 6 sampling sites in Baffin. This enterococci test developed by Adex Industries called EnteroAlert. It's prone to false positives. And the false positives are caused by marine bacteria of the Bacillus genus. So when you're sampling marine water, you need to dilute the water by a factor of 10 with fresh water to basically discourage the growth of those Bacillus species, because you don't want them to be false positives in your test. So it's standard practice to dilute water from recreational marine beaches by 10-fold. Now, I don't like that process simply because if you're diluting to avoid a false positive, you're also risking a false negative on the other end, right? Because you've diluted your sample. So I don't like how that works. It's a little messy. But here's the deal. What's the salinity at a typical ocean recreational beach? 35. What's salinity in Baffin? 50, 55. Right? So diluting by a factor of 10 can cut it. And the salinity in Baffin can change a lot. So for every sample you take, you need to first test salinity and then determine the proper dilution factor. So I don't trust that data, right? Because this was done, we have to run all our samples through a DLAB accredited lab, which was the Noetia County lab. And they did what they're supposed to do, but in hindsight, looking at this, I'm like, well, all of these samples, the dilution factor should have been adjusted according to what the salinity was. So I don't trust any of that data. That column is unreliable, right? All right, so let's move on to the next column. Los Olmos Creek. How high does the salinity get in Los Olmos Creek?
75 or 100 in some cases.
It's up there, right? 75, 80. I've seen it above 100. They had, what was it, last year? There was a halobacterium bloom, an archaea bloom. It turned the water red, right? Because it was this hypersaline halobacterium that bloomed. And that was when it was above 100. And the variability of the salinity in Los Olmos is, you get a tropical storm that comes in, completely flushes it out, it's 0. And then it'll build back up to 100, and then go flushed out again. And it's everything in between. Well, same thing as Baffin Bay. These samples weren't, like the dilution factor wasn't adjusted for salinity. So you can't trust that data either, right? Because these samples are going to be prone to false positives. So the numbers here are probably lower than what's shown on the graph. All right, let's go on to the next one, Petronella. The problem child. As I was looking at this data, I was also looking at land use data. And Petronella the watershed is basically crops. It doesn't have a major population center, right? Kingsville and Alice are in the San Fernando. They're not in Petronella. There's not a lot of livestock in that watershed either. Again, it's mostly crops. There's not a lot of pasture. It's mostly just crops. So how is it so high? And that's when I realized that the sites we're monitoring on the Petronella, even though they're upstream, they're saline sites, right? So what some of you probably are aware of is that salt was a big industry in Laguna Madre a long time ago. There's a lot of salt in the soil. There's salt in the groundwater. There's salt in the tributaries, right? During drought, you can walk along some of these places, and your feet just crunch from all the salt that's just baking in the sun. And there's some saline spots that will some really briny spots that will show up in these tributaries during drought. So E. coli, because we changed colors here, E. coli is the appropriate indicator for freshwater you can't use it in petronella. It's saline. So, we can't trust that data either. That's not reliable. So moving on to San Fernando. San Fernando is actually fresh water. So you can use E. coli. So that column is the only one I trust. The rest of this data I wouldn't trust. And remember, this data mirrors our narrative of how we understand bacterial pollution in Baffin Bay. So I'd like to change that narrative by adding some new data to it, by adding some microbial source tracking data to it. All right. So Baffin Bay watershed. Let me help orient y'all. This is a figure that Dr. Gregory shared from the Texas Water Resources Institute. This was done independently of our EPA study. This was data this was from a workshop that he gave back in 2022, I think. But they looked at land use, land cover, and here is the petronella, and this dark brown color is cultivated crops. So not really livestock there. There's not a big population center there. Driscoll is right here. And here's the San Fernando. We have Kingsville right here. We have Alice right here. Those are our bigger population centers in Baffin Bay watershed. And you can see a transition in the colors here where you get to more of this shrub and scrub and grassland and pasture here in the San Fernando, right? So, again, when you look back at this and you see petronella so high, you're like, well, why? Right? It's just cropland. There's not a whole lot there. All right. So here's our microbial source tracking data. And this graph, it's similar to the previous one, that the scale is log. So when you look at it, understand that these differences, even though they might look small, these differences are actually really big. But the only way to fit these on a graph is to log them, so they all fit. So the differences are actually really big. And Nora and Nicole broke this up into blue for human, green for gull, pink for pig, right? And yellow for cow. And they also divided it by system. So Baffin Bay, our 6 sites in Baffin Bay, our single site in Los Olmos, our 2 sites in Petronella, and our 3 sites on the San Fernando. Am I getting that right? 2 sites in Petronella? Okay. And 3 in San Fernando? Okay. All right. And what you see is that the cow marker is the highest, right? And it's moderate. It's at a moderate height in the bay, and it's a little bit higher in Los Olmos, Petronella, and then it's much higher in the San Fernando, right? That's where we have grazing. That's where we have pastures, right? So that makes sense. Then the pig marker is low in the bay, and then it kind of increases a little bit through the tributaries, and it's highest in San Fernando. Pig, by the way, we're not looking for, the target isn't domesticated pig. The target is feral hog, right? Texas has a big feral hog problem. So we're trying to capture the contribution of wildlife to the fecal pollution problem. And then we have gull, and this marker is confounding. Some of these markers are really specific, like the human marker, the cow marker, the pig marker. These are highly specific for those animals. The gull marker, we call it the gull marker because it was originally developed for gulls. Pretty much any waterfowl is going to trigger this marker. So just think birds, right? And it's important to know what the birds are triggering because there's a lot of birds, right? You know, there's a lot of waterfowl in Baffin Bay, and hopefully, once we dig into the data a little bit more, we'll actually see some seasonal changes in that based on migration, right? Which would be great to see. We haven't looked at that yet, but I hope to get into that. But it's kind of a mixed bag. You know, it's kind of on level throughout. But spatially, it looks a little different. And then we have human. It's the lowest of all the markers, right? And it's lowest in the bay, and then it increases in the tributaries, with its height being in San Fernando. And this is a bit different from the narrative that we've heard about Baffin Bay. We've heard it's human, septic systems, wastewater treatment plants. But for the most part, those systems do the job. So this is what I would expect. Human is the least contributor because when you go poop on the bottom, it goes somewhere, right? It goes to a septic system or it goes to a wastewater treatment plant. But when these other things go poop, it doesn't go to a treatment plant. It doesn't go to a septic system, right? And in Baffin Bay watershed, the 5 counties that make up the Baffin Bay watershed, there's over 100,000 head of cattle, right? It's a lot of cattle. All right, so let's take a closer look at this, or a different look at this. Nicole put together a spatial interpolation of this. So it's the same data. It's this data. But she put it like this. Pretty cool, right? So we've got a map for the human marker. It's a heat map. The hotter the color, the more abundant it is. We've got the cow marker here, pig marker here, gull marker here. All right, and we're going to go through each of these individually, right? So let's start with the human marker. We have some hot colors in the tributaries, and then we get dilution in the bay. That's what we expect. The hottest tributary is the San Fernando. That's where our population centers are, Alice and Kingsville. Kind of makes sense, right? And you don't really see much human marker in the Petronella. All right. So what I always like to ask of my data is, does it make sense? Sometimes data will tell you something that doesn't make sense. So Kingsville population, as of 2023, was almost 25,000. Alice's population was, about 17,500. Right. So those are the population centers in the watershed. So you'd expect that the human marker would be higher there. If you're going to have septic systems that are malfunctioning or wastewater treatment plants that might not be operating optimally, that's probably where it's at, right? And if you look at the land cover map, it makes sense too. So the dark red colors are the developed places where there's a high human population, right? So here's Kingsville here, and here's Alice here. And those are on the San Fernando. So that's where you should see the human marker. You shouldn't really see it in Petronella. It's just cropped in. All right, next is the cow. So the cow marker, you see that you can actually detect it everywhere, right? There's some heat everywhere from the cow marker. There's some in Petronella, but there's a whole lot more in San Fernando. And there's some in Los Olmos. All right. Same question. Does this data make sense? So based on land cover stocking rates and the verification of those rates with the National Agricultural Statistics Service, San Fernando watershed has about 30,000 cattle. And the Petronella has a bit over 8,000. So this makes sense, right? That data makes sense. And the Texas Water Resources Institute, independently, they created this model for E. coli loading for cattle into the watershed. And here is San Fernando. There's Alice right there. There's Kingsville there. Petronella's here. This data is supported by the land use and the modeling of E. coli loading, right? Makes sense. And these studies were independent. I love it when independent studies support each other, because then you're like, oh, we really have something. You know, we can have confidence in this data. So we were really happy to see this. All right, fun fact. Texas, of course, has the largest cattle population in the United States. And the 5 counties that make up the Baffin watershed, there's over 100,000 cattle that reside in this. Now, I'm not saying that all of those are pooping in the Baffin watershed. That's just what's across those counties. You know, in order to see how many are contributing to pollution in the watershed, you have to do these studies like the Texas Water Resources Institute did, where they're looking at land cover stocking rate, they're comparing it with NAS data, and they're talking to stakeholders in the watershed, getting them to verify that data, right? So there's a lot of ground truthing that needs to happen to be able to translate those bigger numbers that you see, like in an agricultural census report, to what might be contributing to the watershed. But fortunately, our friends over at TWRI are doing that. All right, and a mature cow can poop 15 times a day, producing 65 pounds of manure. Cow is a big animal. That's a lot of poop, right? I mean, and that's another thing, right? The narrative that humans are the primary contributor. I mean, we don't poop that much, and we're pooping in toilets most of the time. But a cow, by comparison, you know, right? I mean, the cow's just doing what it does. I'm not blaming the cow. All right, pig market. This one's really fun. We didn't know what we'd get with any of this stuff. You know, we were just throwing darts at the board. We were like, who are the usual suspects? Let's grab some water samples to see who it is. So pig marker. We see some heat right here up in the San Fernando. We see a little bit of heat in the higher regions of the Petronilla. And we even see a little bit of heat in Los Olmos. And we get dilution in the bay. And then we get this weird, like, just little bit of heat right there at where we're exchanging water with the Laguna Madre. I don't know what that's about. I think it's real. And Mike and I have a study that's in review. It's a proposal that hopefully will get reviewed and funded by the EPA, where we're expanding all this work out into the upper Laguna, right? Because I think that's going to really fill in some, close some data gaps for us. But does this data make sense? Well, the Texas Water Resources Institute has looked at feral hog estimates in the watershed. San Fernando watershed, 17,826. Petronilla Creek watershed, 3,933. They didn't do Los Olmos. And Los Olmos, again, it's kind of an anomaly because it's a single site that's in this, really hypersaline tidal section of the creek. But we do have all the land cover data for these 2 watersheds. These estimates are based on 39.4 acres per hog applied to all land except barren land, developed land, and open water, right? And then it's adjusted with a 10% density for cropland. And it's like a best estimate. If you go out in the watershed, it's hard to find them. Like people that work with Texas Wildlife Services, they can go out and do this. They know how to do it. But it's hard for just an average person to go out and find these things. Often you see them, but you're looking at the butt end as it runs away from you up into the brush, right? But those are the estimates. And then they created this E. coli loading model for feral hogs. And here is the Petronilla, and here is the San Fernando. And it's the San Fernando that has the highest load, right? So our data makes sense, right? This independently supports our data for that feed marker, feral hog, which I love to see. All right, fun facts. Texas has the largest population of feral hogs. We're winning at a lot of things, right? 2.5 million of the U.S.'s estimated 6.3 million feral hogs live in Texas. Right? They like it here. And their populations are rapidly expanding. This is a very old source saying the damage estimates begin at 1.5 billion annually, and that conservative estimates for economic damages in the state of Texas exceed $300 million annually. It's much more. That's a bold estimate. But you know, feral hogs are a big problem. They're cute, but they're a big problem. All right. So-called goal marker, which is basically birds, right? So this is the one that is, it's got some spatial distribution we need to pay attention to because it's everywhere, right? The birds are everywhere. Yes?
I thought of a question about the hogs. Would the marker also count javelinas?
It would. It would count javelinas, and it would also count the birds. Yeah. So it's not. It'd be really hard to get a marker that was so specific. It was just the feral hog. You know, but the javelinas. I think what we're measuring is there's some contribution, and there's some contribution from domestic pigs too, but it's the smaller end of the contribution. But you're right, there is cross-reactivity there. And in this marker, there's a lot of cross-reactivity. There's a lot of bird poop that'll trigger this marker. So, but it kind of makes sense once you think about it. So, birds are going to congregate and roost in the tidal sections of these tributaries. That's where you're going to see them the most. If you're motoring across Baffin Bay, you might see some migratory birds overhead, some ducks, some geese, or something, but you're not going to see them all just sitting out in the middle of the bay, right? So you're going to get a higher concentration along those tidal sections where they're going to roost, and you're going to get a dilution of that out into the bay. But this makes sense because Baffin is home to a large population of waterfowl. There's a lot of waterfowl that live in Baffin Bay, that migrate to Baffin Bay. What I'm wondering is, this is the spatial interpolation of the data. I'm wondering about a seasonal interpolation. Because if we can match these concentrations to migratory patterns, then I think we have a deeper story. So that's something we still need to look at, which will be really fun to do. All right, so what are our conclusions? The dominant fecal sources in Baffin Bay are, in order of magnitude, cattle, birds, pigs, and humans. All right, so our narrative for Baffin is changing, which is, we need to adjust it. As we get new data, we need to adjust our narrative so we can prioritize our management strategies. San Fernando Creek is the most fecally polluted tributary. So again, we're changing the narrative a little bit. We've long been considering Petronilla to be the most fecally polluted tributary because we've all been using the wrong bacterial indicator for Petronilla. It's saline. And the land use data doesn't support that at all. For this, the microbial surfactant data, it's got strong support from land use data, livestock data, and wildlife inventory data, which is really good to know. And these results add to the story of what we know about pollution in Baffin Bay, and it'll help us prioritize our mitigation strategies to protect Baffin Bay, to bring Baffin back, right? And that's why it's so important to have Lucas Gregory and others at the Texas Water Resources Institute working on this project with us, because they developed the watershed protection plan, and they can make this data actionable, right? So that'll be one of the next steps. One of the next steps is the quantitative microbial risk assessment, and the other step is incorporating this into the watershed protection plan. All right, and we had a couple great questions through the presentation, but what other questions do we have? Travis?
Yeah, it's about the dominant animals. You had a lot of different comparisons. Some made more sense than the others, right? Now it's one river, now it's the other. Casa here, casa the other. But the question I have is, all poop created equally. Is there possibly more bacteria per gram of cow poop versus human poop? Humans are doing a lot of antibiotics and stuff, so maybe they're big so much bacteria, 10 times as much poop for the same amount of bacteria.
That's a great question. All poop is not created equal, right? A cow is going to have a very different microbiome than a pig, than a human. So that's a good question. I don't know how that translates to the microbial source tracking data, but that's a good question. And just, fun fact, E. coli, the indicator that's appropriate for freshwater, it's a gram-negative bacterium. Enterococci, the indicator that's appropriate for marine water, is a group of gram-positive bacteria. In the bacterial world, genetic diversity is huge. So you are more similar genetically to a potato than a gram-positive is to a gram-negative. Right? So these poop bugs aren't created equally either. Right? So that graph I had where we had both E. coli and enterococci on the same graph, that makes no sense. Right? Yeah. Fun fact. Yes, Colleen?
All range, or is there any defined in the operations? Is there any land applications, cattle?
Well, that's kind of the next thing we need to learn, right? We need to work with the stakeholders in the watershed, and Lucas Gregory and the TWI have already been doing that. But we need to see what the best management practices are that are in place, and kind of reevaluate what the strategies are for the bay. So that's the next step, right? We've got this fresh well. Now we need to make it actionable. Adrian?
So I think, the watershed protection plan obviously identifies management measures that across all of those bacteria sources.
Yeah.
And so I think, with this, there certainly needs to be more of an emphasis on your cow inputs, to say the least. But on the topic of poop not being created equal, can you talk a little bit about the risk assessment, the next step here, the risk assessment and how that ties in, or how that affects kind of our understanding of your ongoing risk?
Yeah. So the risk assessment is potentially the more important part of this study, and that's ongoing. Dr. Anna Gitter at UT Health and Dr. Nicole Powers, who's now at the Hart Research Institute, are working on that, and they're asking, well, what do these numbers mean? What risk is posed to you as a recreational user, right? If you're recreating in an area where bird fecal pollution is very high, it's really not that big of a deal, right? It takes a lot of bird fecal pollution to have the same risk as human fecal pollution. Human fecal pollution can have the bacteria and viruses and parasites that make you sick. The bird, probably not, right? Cattle, that's going to be almost a 1-to-1 for humans, though, in the risk assessment. So that's a big concern when we start doing the risk assessment. I'm really interested to see what the risk assessment models show us with the cattle fecal pollution. Because the literature I've seen is that in terms of risk, it's almost a 1-to-1 between cattle and human. And the pig, there just haven't been a lot of risk assessments on pheromones. So I think there's going to be some creativity in that modeling. We'll see what happens. But I've seen the microbiome of domestic pigs and wild pigs, and I'd rather be dealing with the wild pig microbiome. Carol, you okay? Yeah, so the risk assessment is going to be a big part of this. And I'm really looking forward to seeing what that data looks like. I don't know how to do risk assessment. That's all Anna and Nicole. Yeah. Other questions?
Yeah, you asked where you talked about the temporal qualities of some of this data, and I was curious if you had a timeline for doing that. And I was really interested to see, especially for cattle, if that lines up at all with like big livestock shows in the area. It's something our stakeholders have talked about too, and
Yeah.
I'm just excited to see that and to learn more.
Yeah, right. That'd, you know, it'd be really interesting to see, if we knew the dynamics of what's happening with the cattle populations, and we could look to see what overlaps there are between those data sets. That'd be great. I don't know how to get that data. You know, you're on the ground with Baffin Bay.
We'll work on it.
You might know how. Lucas might know how. That'd be really interesting. Ironically, the graph I showed y'all that I stole from Nicole. Let's see. Let's find it real quick. Oh, this one right here. All right. So this big hotspot of fecal indicator bacterial pollution in Matagorda and Brazoria. Guess what 2 counties on coastal Texas have the highest cattle populations? Matagorda and Brazoria, right? A lot higher. Yeah. I think there's something there.
Go back to the graph again.
Which one?
Sorry, that one you were just on.
Oh yeah.
You also mentioned that sometimes some of the bacteria doesn't survive nearly as well in saltier conditions. And
That's right.
On the coast, we get saltier and saltier.
Yeah. Yeah. So the water here, the coastal water up in Matagorda and Brazoria, is not going to be hypersaline. But in the Baffin Bay system, it usually is, right? So those fecal indicator bacteria, they're happy living in a mammalian gut. It's a lot saltier in Baffin Bay than mammalian gut. So the life expectancy of those bacteria once they get out in the bay isn't very long, which is good. You don't want an indicator that'll get in the bay and then grow again, because then how can you measure pollution, right? Because then it's naturalized to the bed, right? So a hypersaline system might be a better system to use a fecal indicator bacterium because its growth is going to be more limited. Whereas in a more freshwater system, it can regrow, right? It's important. Yes?
So I know in the Bringing Baffin Back initiative, a lot of the focus has been on human source pollution. So what is different between the way that whatever data that's founded on was collected and how it was done here?
Yeah, well, that's fecal indicator bacterial pollution. So that is this type of data. And this data mirrors that historical data. So a lot of our narrative about Baffin's fecal pollution is driven by data that looks just like this. But for various reasons, that data is not reliable.
And so when you use this, there's really no way to track the source. It's just an assumption of where it's
Yep. It's anyone's guess. Yeah. And in any watershed, humans usually get the blame. But I don't like to blame humans because you're usually blaming wastewater treatment plants and septic systems. And a lot of times, they're operating optimally. There are malfunctioning septic systems. There are suboptimal septic systems. And there are wastewater treatment plants that aren't operating optimally. But they're all doing their job. Right? And just the sheer numbers. It's a rural watershed. There's not a whole lot of people that live in the Baffin Bay watershed. Right? You can't have this huge fecal pollution problem where you don't have people. Sam?
So with Petronila being, it's brackish, and it's, a lot of it's from historic oil and gas, brine.
Yeah.
And so with that being, brackish and that, 15 to 20 parts per range, not hypersaline, not oceanic, but still elevated, do you think that maybe switching to an E. coli as an indicator, the interim outsized indicator in that?
Yeah, it would be the more appropriate indicator for that system. And again, because of salinity range, you'd want to test the salinity to determine your dilution factor first.
As a temperature effect, did you say that you have done the annual analysis or that you're projecting out to do throughout the year kind of assessment? Or did you show it and I didn't see it?
I didn't show it here, but warmer water temperatures are typically correlated with higher fecal indicator bacteria.
So we would then expect to see a summer.
Yeah. Yeah. For the mild seasonality we see here, there'll be a little bit of a seasonal trend. I'm really interested to look at that. I want to talk to some bird people and figure out what the relations, how this data compares with bird migration. Adrian?
Did you, especially, so in talking about the cattle here, in the tributaries, we can say San Fernando specifically, did you see any variability based on rainfall? Was it, consistent, elevated concentration throughout the year between sampling events? I ask because I know one of the management measures for cattle is to prevent them, or block off access to the creek as a way to help reduce the flows. And so,
yeah, that's a great question. So what's the variability with rainfall? Well, Nora created this amazing figure that looks at the microbial source tracking marker abundance with rainfall. What's that? Oh, this one doesn't have rainfall?
Well, because I separated the weather stations based on the list and maybe some working on a better way.
Oh, gotcha.
Patrick, you had
So we're working on that.
Yes.
We're working on that. Nora's all over it. Yeah, right. So there's different management practices related to rainfall. I expect we'll see some strong rainfall trends, but I don't want to get too excited about that. They may be diminished because of we didn't have a lot of rainfall events during those 18 months, not significant ones anyway. That's one reason why we went back to the EPA to not only expand this to fill data gaps in the upper Laguna Madre, but to capture more wet weather events, so we can better understand what happens when it rains, right? Yeah, that's a good question. Yes?
Do you think there could be any potential to mitigate the cattle runoff with like bioremediation, like planting plants? Would that help?
Riparian buffers are great, right? Riparian buffers work fantastically, and I mean, just letting creek banks go wild is a great way to have a riparian buffer, and just minimizing the amount of time livestock can spend next to the creek, right? That's a great way to do it too. I think that's an easy, best management practice. And I mean, the cattle ranchers, they know how to do this stuff. Yeah, I think that's that. And all this stuff is so fixable, right? We can fix all of this.
There are some efforts being made to try to do that. I'm working on one at Petronila Creek, trying to expand the buffer to do exactly what you said to do. So expand the buffers and hopefully get a little green space to absorb some of the pollutants. There's some money out there, hopefully. We'll see.
Are we still assuming that there is a deleterious impact from these bacteria, or did you say we still don't know what the impact is in other places?
Well, there's a risk that's posed to human health, right?
We know that.
But that's that quantitating that risk, like measuring that risk, calculating that risk is the next part of the study, right? So that's the next part of the study.
taking human volunteers.
No, we don't do that anymore. There was a time when postdocs and grad students were volunteers. Those are we're decades past that. I've heard stories.
Oh, I just wanted to hop in and add to that. I noted about riparian and stream bank restoration. We, in the past year, through that watershed protection plan that Dr. Turner mentioned, had worked with producers in the watershed to begin to implement some of those practices. And I think your data is just going to beef up that work there and hopefully bring more money in to implement the stuff that we've covered. Almost, I think it's about 1,200 acres so far of land that's been kind of, again, beefed up with these, literally beefed up, I guess, best management practices to keep cattle out of the stream, to provide them an alternate water source, and hopefully start replanting some of those areas and bringing those nutrients and bacteria out of the water that you're planting. So we're really excited about it, and again, hope that this will continue to bring more interest to that topic. Super cool.
Yeah, it's really cool to watch all these collaborative efforts that are happening in Baffin and see, the data adding up and the practices adding up and kind of seeing this trajectory for, the reality and probability of bringing back. It's a lot of fun to see it all and be part of it. I want to put this slide up. I'm looking for food, and maybe y'all know a source. I need some nilgai food. It needs to be fresh and it needs to be refrigerated. Okay? So if anybody can get me some nilgai poo, I need it. But I see some hands. You traffic in nilgai poo?
I know it.
Yeah. As my friend says, freshly voided pieces. Yeah, exactly. Honest. Because we don't know, there's no studies that have run for cross-reactivity with cow marker and nilgai. This is the only watershed that the questions ever come up. So we want to know if the nilgai if the cow marker also is positive in nilgai pieces. No, I'm asking. No, I'll drive to you. I'll pick it up. Oh yeah? Perfect. Yeah, or I can sneak down there, just show me where it is, right? Yeah.
How many nail guys are there compared to cows?
It's not really comparable. But, just like understanding that javelina are cross-reactive to the feral hog as our domestic pigs, I think we need to resolve out all the cross-reactivity. We really need to, focus on the details at this point. Yeah. Great. Thanks, everyone.
Fantastic, thank you.
Did I take up the whole time?
Yes.
Oh, yeah. 7:20, I didn't realize it was that late. I wasn't planning on speaking that for that.
Yeah. Oh, that's all right. No, thank you.
See you in March.
Yes, March 3rd is our student sit. So we'll have college students from Texas A&M, Kingsville, Corpus Christi, Del Mar, and UTNS High. So we'll hear about their cutting edge research projects and hopefully, inspire more folks in our community. All right. Thank you guys very much.
Thanks, everyone. Have a good night.
Have a good night.
Oh, do we have a right?
Oh, yes. Oh, my God.
Yeah, we need to put them on. Otherwise, it's people. They like it.
All right.
Raffle 222 312.
And there's Good job. Yeah, yeah, yeah. Okay.
My favorite place to go. It's never
Good job.
I love it.